from doing the work of, from the angle of, okay, how is this trait going to get passed down? You know, what likelihood do you have of getting this trait? You know, what are the odds you're going to go bald? To looking at how traits get moved through families. Now we zoom in on, well, okay, if it's the DNA part, if it's the gene that was making that trait happen, how did that DNA actually get that done, right? Okay, we know DNA gets passed on in certain ways, but how does it actually make your hair brown? How does it actually make your eyes green? How does it make you tall? And the simple answer to that, we already know. What is it that DNA does? What does it code for? DNA codes for protein. That really is at the heart of these two chapters, how DNA makes proteins happen. So that's where we're going, but it's a long answer. First of all, how much DNA do you actually have? How much is there? Let's take one cell, because we know that every cell in your body except for gametes has the same set of DNA, right? It's the same amount in each nucleus. How much DNA is there actually in one human nucleus? Let's go with length. Like if you took that DNA, this stuff, right? This is DNA. If you took it and unwound it into like single, double strand, how long would it be? You took all of them, maybe, probably more than that. But just one, no, just one, and you stretch it out. It's about 6 feet. About 6 feet of DNA. Of course, multiply that by 100 trillion, because that's how many cells you have. And now we have 600 trillion feet, which is, I wish I was better at a sense of universal scale. It's probably to the moon and back. I don't know. It's able to do that because of the kind of packing that you saw, right? It winds around those histone proteins. It folds up into 5 different levels. It coils at 5 different levels to make a chromosome. But even in its normal chromatin state, it's got 2 levels of coiling. DNA chemically is a nucleic acid. It's our 4th group of macromolecules. Remember, we did carbohydrates, lipids, proteins. Nucleic acids were the ones we kind of skimmed over because we know we're going to hit them hard here. What our smallest unit that we can still call DNA is called a nucleotide. So one nucleotide of DNA has 3 components to it. Deoxyribose is a 5-carbon sugar as opposed to the 6-carbon sugars you're used to from biochemistry, right? C6H12O6. Deoxyribose is similar, but with only 5 carbons. In fact, deoxyribose is basically a sugar called ribose without an oxygen, missing 1 oxygen, hence the name deoxyribose. If it had that oxygen, what molecule would we be building? Maybe it's worth writing the full name of DNA now, which is, let's go green, deoxy. Deoxyribonucleic acid, that's what DNA stands for. DNA. So if it was ribose in this spot, what would we be building? Oh, 3rd period. You are sleepy. DNA is deoxyribonucleic acid because it has deoxyribose as its 5-carbon sugar. We'd be making RNA if we were using ribose instead of deoxyribose, and we'll get there. There's always a phosphate, which is vaguely spherical. And then the code part. You know, DNA is a code. DNA is a code because in the third spot, turns out there's 4 different things. There's purines, 2 of them, called adenine and guanine, and there are pyrimidines, 2 of them, called thymine and cytosine. The purines are double ring structures, 2 rings of carbons, with nitrogen thrown in there a couple places. The pyrimidines are single hexagonal rings. And by putting them together the right way, I mean, the right things have to be bonded to the right things. And at this point, we are talking about covalent bonds, electron sharing type bonds. In one nucleotide, everything is covalently bonded to everything else. So one nucleotide, the smallest unit we can call DNA, looks like this. We got the phosphate bonded to one end of the deoxyribose. We have the nitrogenous base bonded to the other end of the deoxyribose. And in fact, if you chain phosphates and deoxyriboses together, like so, that would be one strand of DNA. Questions so far? Okay. Right. What changes from nucleotide to nucleotide of DNA is simply what's in that spot, right? It could be any of the 4 nitrogenous bases, adenine, guanine, thymine, or cytosine. Any of them. Now, I've only got 2 there because I was copying and pasting. I didn't want to do it again. But it could be all 4, right? And it turns out that the sequence of those 4 nitrogenous bases, nitrogenous just means nitrogen containing, by the way. The sequence of those 4 bases is the whole code. Like, that's what makes this thing give you brown eyes and your height and certain aspects of your immune system and so on. And if that doesn't sound like enough, if 4 different nitrogenous bases being in a different order doesn't sound like enough of a code to create you, keep in mind that the most complicated computer in the world, I don't know what that is, whatever it is built on a much simpler code, binary, which is just zeros and ones. Here we have 4 possibilities, exponentially, more complex, which is why, in fact, a lot of people think the future of computing is going to be biological computing, using biological molecules to store information. To talk about DNA, we do have to talk about Watson and Crick. Watson and Crick being the 2 scientists who were, and still mostly are, credited with discovering the structure of DNA. By all accounts, not the nicest of guys. Basically, they were stuck at the, well, kind of where we're at, right here. They were kind of stuck there. We'd gotten beyond T.H. Morgan. T.H. Morgan discovered, hey, chromosomes are the thing, right? Chromosomes are the things that are coding for the stuff in fruit flies, so they must be in humans as well. And then somebody after him figured out, hey, it's DNA in those chromosomes, not the protein part, it's the DNA part that is the code. And here they are. Okay, we know what DNA looks like chemically, at least one piece of DNA. We analyzed, we know it's got the deoxyribose and the phosphate and all those nitrogenous bases, but we don't know how it all fits together. They didn't know this, right? They didn't know this twisted ladder looking thing. And that's where Watson and Crick are stuck with these ball and stick models of chemicals until something happens, which is that they're stuck and they call their buddy Maurice Wilkins, who works in a lab across the country. And they say, hey, Maurice, we're stuck. We don't know what to do here. We need some fancy pictures. We know that you have some people at your lab, especially one person who's really good at X-ray crystallography, this way of taking pictures of molecules Instead of using light, it uses X-rays. And Maurice is like, well, yeah, I mean, she's really good at it, but like, she doesn't like me at all. Like, we don't get along very well at all. Turns out, like, he kind of acted like he was her boss, and she didn't think. Anyway, office politics type stuff. But the upshot is that Maurice basically got into Rosalind Franklin's stuff. Rosalind Franklin's the X-ray crystallographer and scientist. Got into her stuff and stole a picture. The picture, pretty famous now. This, and what that is basically this thing, but looking down the barrel of it. And that was all Watson and Crick needed. Basically, and they saw that and went, oh, okay. So the nitrogenous bases go in the middle, and it's two different strands, and they must be going different directions. You'll see when we get out our paper shapes. But basically, her name needs to be mentioned here because they won the Nobel Prize for this, and she got no credit at all. They called it the double helix. Helix means a coil, kind of spring shape. They discovered there was actually two strands of DNA twisting around each other. And if this only works, the chemical structure of DNA only works, they only fit together right if the strands run in opposite directions. And the word they used for that is anti-parallel. That a DNA strand has sort of a head end and a tail end, for lack of a better term, and it only works if they run like this, if they run in opposite directions, twisting around each other in a coil. In the middle of that coil is where the nitrogenous bases are, and they are facing each other. And in fact, they are bonded by a different kind of chemical bond than covalent, which is the kind where electrons get shared. Weak hydrogen bonds is what the bonds in the middle of the DNA molecule are called. They're called that because they are weaker than covalent bonds. And in fact, the fact that they are weaker than covalent bonds is part of the key to how DNA is used. It's very easy for an enzyme to split, to unzip the double helix and get at the code. Because of the work of another scientist named Chargaff, I think Erwin Chargaff? Anyway, Chargaff's rule, basically, before Watson and Crick, he had measured that there's always the same amount of the nitrogenous base, okay, A, T, G, and C, right? Adenine, thymine, guanine, cytosine. Chargaff measured that there's always the same amount of adenine that there is of thymine in a cell, and there's always the same amount of cytosine that there is of guanine. But what Watson and Crick figured out from that is that's the pairing. In the middle of the DNA, adenine's always bonding with thymine, and guanine is always bonding with cytosine. That might be the most important take home of the day. You're going to need to memorize that quite quickly once we get rolling here after the test. Luckily, I'm all about stupid ways to remember stuff. Here's how I remember that adenine always bonds with thymine and cytosine always bonds with guanine. When I write those capital letters, I immediately notice that A and T are made of straight lines, and C and G are made of big curvy lines. Literally, that is how I have remembered those pairings for 30 years. That works for you, great. If not, use something else. Not in DNA. So we don't get U until we get to RNA. So we'll talk about that when we get there. Promise.
So the A, T, G, and C, like on the model, they're always facing towards the center.
Yeah, so like if you look down this thing, G is always across from C, A is always across from T. Yep. Because that's how their shape allows them to bond. The A and the T are able to form those weak hydrogen bonds with each other because they stick the right charged parts out so that they create that attraction. And then the C and the G, same thing, right? They don't bond apart from that parent. Questions? Good job. Tomorrow's your test over 11 and 14. I do have guided reading for you for chapter 12. It's those easy ones though. Not a whole lot of deep questions. It's due. Oh, what do you think? Friday?